second of our 3 lectures on microbial evolution. And today we're going to talk about, we're going to come back to the topic of bacteriophage and specifically talk about how to use lytic bacteriophage as a way to treat infections. And this is kind of an area that is becoming popular. So just to kind of give you a bit of context, in the 1920s or so, before the identification of penicillin, phage were thought as being kind of this revolutionary miracle, quote-unquote, drug as a way to treat infections. So there were studies done, particularly in India, in the 1920s using phage that infected Vibrio cholerae using phage that infect Vibrio cholerae as a way to prevent cholera infections. And it worked pretty well. Now, study designs, clinical study designs in the 1920s are completely different than they are now, and so once penicillin became more popular, with the ease of producing penicillin and with its ability to target a broad spectrum of different microorganisms, it was thought that these antibiotics would be the preferable method in the long run. So Western governments decided to put money into antibiotics. Eastern European countries actually kept money in phage research. So if you look at where this has progressed the most, actually Russia has some of the most, probably the greatest expertise in using phage as a way to treat infections. These days in Russia and Ukraine and some other countries, you can actually buy phage over the counter as a way to treat skin infections, primarily staphylococcus infections. But there's been some renewed hope or renewed, I suppose, a push to use phage therapy in the U.S. Reason being, antibiotic resistance. As microorganisms become more, as the percentage of organisms that become resistant to our best antibiotics become more common, what we're finding is that individuals are looking for new and novel ways to treat infection. And bacteriophage represent one of those mechanisms. So the very crude and elementary phage research or phage therapy methods would be ones where you would basically provide somebody with a cocktail of phage, all treating one particular bacterium, E. coli for example, with the goal of being to kill off that bacterial species. Okay? What's the benefit here? So what is the benefit of this relative to using an antibiotic? What are the downsides of antibiotics? Let's start with that. What's that, Emma? You can build up resistance, and actually we don't overcome that necessarily with phage. There can be resistance to phage as well. Actually, in our lab, the students that work with me, we can detect resistance within 24 hours. Okay. What are other reasons? And it kind of depends on the concentration of phage that you're using. What else do we know about bacteria?
Oh, well, antibiotics can also damage healthy cells as well.
Good. So there's collateral damage is the way that we would describe it. We looked at an example where in using oral antibiotics, broad-spectrum antibiotics can lead to the rise of a C. diff infection. You wipe out the good or natural microbiome of the individual. That allows for opportunistic infections to take place. How are phage an advantage in this idea of broad and narrow spectrum? Really comes down to phage are incredibly specific toward their host microbe. So for example, phage that infect E. coli, a lab strain of E. coli, they will not infect Salmonella, they will not infect Lactobacillus or Bacteroides or the Enterococci that help to colonize the gut. They will only go after E. coli. Okay? In the same way that in the US, or sorry, worldwide, we have diseases like Epstein-Barr virus, influenza. They don't infect all animals. Those are ones that are primarily specific to humans. Kind of a broad overview. And this is kind of a new venture of phage therapy in which the investigators, and this comes out of Yale, they were looking at a bacteriophage that they isolated just out of a pond. And they found that this bacteriophage targeted a multi-drug resistance pump known as OPRM. Many of you identified correctly, these efflux pumps are a mechanism of resistance. The goal of these efflux pumps, or how they function, is antibiotic gets into the cell, uses active transport to just pump it right back out. Okay? The OMK01 phage, so this Pseudomonas phage, binds specifically to OPRM. That binding is required for the attachment step, the first step of the viral life cycle. And after that, we get the viral penetration, where it inserts its DNA into the host cell. We get biosynthesis, assembly, more virus to come out. What they noticed, though, was that some of the bacterial cells gained resistance to that phage within 24 hours. And one of the things that they explore is how do those mutants differ from the original wild type or parent cells? Okay? So that's what we're going to take a look at today is what were these effects and how are they using that. But before we even get to that point, mutation of the receptor that the phage binds to, that's going to block the step of attachment. Therefore, it's going to block every other step of the pathway. So they subdivide in this paper that we'll take a look at pieces of, they subdivide into what they call phage-sensitive bacteria. Those are kind of like the parent strain before being exposed to the phage, and then the phage-resistant. We'll talk about where those come from a little bit later. Okay? All right. So I think we've looked at this a little bit, but this image on the right here is what a plate looks like after we've taken, for example, E. coli and put it onto a plate where we've mixed it with some water, and in that water we can find multiple different phage. Each of these here is known as a plaque. What is a plaque? What's that? No growth. Why isn't there growth there? Not resistant. Those spots where we've got no growth are actually because the bacteriophage have done something. What's happening in these spots, or what has happened to lead to these failures?
Are they rupturing?
Yeah, exactly. We've got cell lysis. So if you think about the steps that are taking to get here, again, we look at the steps of viral replication. First step is attachment. Phage attached to the host cell, penetrate their DNA into the host cell. Third step, biosynthesis. Four, assemble. 5, mature, and then get out as soon as possible. In E. coli, again, with good E. coli phage, this can happen within 20-30 minutes. That process is as fast, if not faster, than the life cycle, or the amount of time that it takes for one bacterial cell to become 2. So this zone of clearing that we see here, or this plaque, is a result of lysis or rupturing of those bacterial cells. And if we were to take, for example, a toothpick and just pick this plaque here, what we'd find is that there are probably millions of bacteriophage within each of these spots. Okay? And each of these spots then represents an individual phage that's replicated and infected those cells. All right. So what we're going to do through this is I'm going to walk through parts of it with you, and then I'll have you guys break out at certain points too. But next up here, so they isolate this phage. They call it OMK01. Chan, who is the primary researcher on this paper, and then Paul Turner, whose lab this came out of, what they ended up doing was testing for the ability of OMK01, the phage, to infect Pseudomonas aeruginosa that lacked the OPRM gene. Okay, so it has a deletion that completely knocks out that particular gene encoding that protein. Good control would be to test another deletion mutant to make sure that the effects they observe are not the results of generating a mutation but rather because of the OPRM gene itself. So, what I want you guys to do is envision what would these results look like. Would the phage be able to infect wild-type Pseudomonas aeruginosa, one that lacks the OPRM gene, and then one that lacks another gene known as I think it's OPRU, is that right?
OMPU.
Thank you, OMPU. So would it be able to infect wild-type, an OMPU deleted gene, or one with lacking the OMPU deleted OMPU gene, or OPRM? That's what I'm getting at. So, predict the results. Okay? I'll give you a minute to think about this one. So, excuse me. OMKO1, phage that infects Pseudomonas aeruginosa. If we take Pseudomonas aeruginosa in this phage and mix them together, plate it out, what do we expect to see? What would we observe? Are the phage capable of infecting wild-type Pseudomonas aeruginosa, one that has all the genes of the bacteria? And the answer to that is yes, they can. So what would we observe? What would these look like on a plate? Think back to the last picture we saw. Taylor, what's on that previous picture? Yeah. Yeah. So we would have a lawn of bacteria, but if the phage is present at any of those locations on the lawn, we get these plaques, these areas where there's no bacterial growth occurring. Okay? And again, plaques just represent that the phage has undergone multiple replications in the bacterial cells that are at that location. Okay? If we delete a gene like OMPU, which is not a receptor for this bacteriophage, what would we expect to see? Why? Or another way to say it is it's not required for the viral life cycle, right? So we'd expect, as Molly pointed out correctly, we'd expect to see the exact same phenotype. If the gene is not essential for replication, then with it or without it should have no effect. on the life cycle or the replication of the virus. Now what if we knock out that receptor that's required for attachment? Will we see infection occur? No. If you knock out any of those steps of the life cycle by deleting essential host genes, for example, if you knocked out the host ribosome, you're not going to get translation of the viral proteins, therefore you won't get new viral particles formed. So we'd see no plaques in this last case here. Inability to infect. Okay. So these would represent then ones that are resistant to our bacteriophage, and these over here would represent sensitive. Okay. Now let's take a quick step back. Why even go about doing this experiment in the first place? Why should they care about identifying a phage that infects Pseudomonas aeruginosa? Does the paper that I had you guys bring up anything about Pseudomonas and why this is important? What do we know about Pseudomonas? Anybody know anything about Pseudomonas? Okay. A couple of quick things, just so you know the importance of this. One, Pseudomonas aeruginosa is resistant to many antibiotics. Part of the reason? Efflux pumps, like OPRM. Other reasons? It produces biofilms. Biofilms are going to be, bacteria that are found within biofilms are oftentimes more resistant to antibiotics than bacteria that are free-floating. And because Pseudomonas can form biofilms, not only does it make it resistant, but it also makes it really challenging to remove. Individuals who have cystic fibrosis often have infections caused by Pseudomonas aeruginosa because they can't clear the Pseudomonas from the airwaves. And because of that, Pseudomonas can colonize, can form biofilms within the lungs, which makes it then much more challenging to treat. It also makes it actually very challenging for the immune system to combat or to defeat Pseudomonas, because it makes it pretty hard for those cells to be phagocytized and broken down by white blood cells. That's something we'll come back to in about a week or so. All right, so now I want you to think about the next step of this process. So repeat the experiments. What they ended up doing next, likely, is to measure the growth of Pseudomonas aeruginosa with a spectrophotometer. Process that, or a technique that we've used before as well. And they're going to do this in the presence and in the absence of this particular phage. Here are some quick notes. Pseudomonas has a 1-hour log phase. KO1 was added to the culture 3 hours after growth began. And that's about it. So you have two conditions. One with phage being added after 3 hours. The other, the cells are going to be growing as normal even after 3 hours. What I want you to do is map out for me, what do you think the growth curves are going to look like with and without phage? Make sure to identify those stages too. Okay, that's a good review for final, which is only 4.5 weeks away now. Yay! But does it look like it? No. All right. So go ahead and begin to work on this one. Take a little bit of time. Feel free to work with a partner on it. All right. So yes, the word problem is not best. Apparently I need to go back and fix this over the summer. But key takeaway points here. Most important thing, I think you can break this down into a very simple problem where you have two conditions. One, untreated, no bacteriophage, just normal growth of Pseudomonas aeruginosa, and then one where you do have a treatment, which is addition of phage. So if we were to draw this out, actually, scroll up a little bit. Okay, there we go. So I'll draw my untreated in purple. 1 hour lag phase. And you can start this anywhere on the OD reading. The importance is not where does it start, the importance is more the general trend here. Okay? So 1 hour lag. Then we've got a log phase, and I just figured I'll take the log phase out to 8 hours. And at least we're going to see stationary. We may see death at that point too. Okay? So this is no phage, or phage if you're French. Now, let's do another one with phage. We're going to see the same thing for the first 3 hours. Okay, we're treating them the exact same way for the first 3 hours. But then we add bacteriophage. What's going to happen at this point? There's going to be death. Phage are going to infect those bacterial cells, produce more virus, and ultimately lyse them. And that's going to lead to a decrease in the optical density, a decrease in the amount of bacteria. So we might see something like, zoop, and then just a flat line. That's a good prediction. It's not the only one, but that's a good prediction. Let's take this one step further real quick. What would happen if we tested this same exact condition? We had phage, but our strain is one that lacks or has a deletion for the OPRM gene. So it lacks the receptor. What do we expect to see? Which of these two curves would it look most like? Why?
Because it doesn't affect the viral life cycle.
If you don't have the receptor, if the receptor is not there, you can't get attachment, therefore you can't get viral life cycle. So yeah, that won't, this last option here would look a lot like just the no phage condition. Okay. Did anybody flip ahead to the next page?
Yes.
That's the answer. All right. Bastard. All right. So this is some sample data here, in which exact conditions that we were just talking about. So we've got an hour-long lag, couple of hours of log, add phage at 3 hours, and then we start to see divergence at that point. So the blue represents no phage, the orange represents when the OMK01 phage has been added. We see a little bit more of an increase, and then this plummet, and it stays low for a while, and then it all of a sudden starts to increase again. What's happening here? Why do we see an increase in the amount of bacteria all of a sudden? Those are the ones that are resistant to phage. Exactly. So in essence, what we have here, if you want to think about this through evolutionary theory for a minute, evolutionary theory is that there's some sort of selective pressure that drives selection for mutations. The phage serve as the selective pressure. The mutations, at least the ones that they identified, are all within OPRM. They're all within the receptor, and so they're blocking those initial steps of attachment and preventing then the phage to be able to infect these cells any further. So these become our phage-resistant population. And our phage-sensitive would be those at time point 0. Okay. OPRM's a multi-drug efflux pump. We've got a mutation. Let's assume it's a missense mutation that has rendered them resistant to the phage. What else might we expect would be the effect of this missense mutation? And a lot of you noted this on the third one. Mutation to a multi-drug resistance pump is going to do what to the bacterial cells? Will they become more resistant or more sensitive to antibiotics?
probably more sensitive. That's what we might expect, right? If you mutate the efflux pump, that means that what you're hoping for, or at least what they were hoping for, is that means you mutate the function of it to pump out those antibiotics as well. That captures the antibiotics inside of the cell, therefore takes less to kill Pseudomonas aeruginosa. That was the hope. Okay, that was the hope. So here is some data for that. So this is looking at just one antibiotic, tetracycline. Tetracycline targets the ribosome. And here are a number of different strains. PA01, PA14, and PAN. The PA stands for Pseudomonas aeruginosa. These first three here are all just laboratory strains that are commonly used to study Pseudomonas aeruginosa. The next two here, the ones with numbers, are ones that were environmental isolates, I think actually from a hospital sink drain, if I'm not mistaken. And then the last three are actually all from patients that were experiencing Pseudomonas infections. And actually, if I'm not mistaken, one of these three, I can never remember which one, but one of these three is from the patient that was talked about in that article. Okay, so I actually tested this on on the strain that was causing his infection. So what I want you to do real quick is take a look at the two populations here. What they do is they average what is the minimal inhibitory concentration that it takes to inhibit individual clones or individual cells of phage sensitive or phage resistant mutants. I want you to compare these two to each other and look at, for each of those strains, what's the general trend here? Okay? Try to describe what that trend is, and then once you have, interpret the meaning of it. It'll take about 3 or 4 minutes. What is the general trend here? How do mix compare between the two groups? Okay, so Emma said that those that are OMK, the strains of bacteria that are OMKO1 sensitive, so they're still sensitive to the phage. You've said that in general these are higher than the mix for the resistant strains. Okay, everybody on board with that? And we could calculate this change or this difference. So what they do is the researchers actually set up a ratio here. Let's see how much of a difference there is between these mix. And if we just do this by hand, well, the sensitive, it's about 3-fold higher than the resistant. About 3-fold higher, 1. Sorry, now my numbers are getting really bad. 5. Roughly 3 to 5-fold difference. In other words, it's taking 3 times to 5 times more antibiotic to kill those sensitive, OMKO1 sensitive, those that are phage sensitive, versus those that are phage resistant. Why? What accounts for this difference here? And again, think about what's the target of the phage and what the function of that target is. So what are the phage binding to? What protein? Come on. What's the receptor it binds to? This phage is binding to what bacterial protein for the attachment step? Not the ribosome. The efflux pump. When it binds, if So what is this saying then? These mutants we know are resistant to bacteriophage. They've got a mutation in the efflux pump. What does that mutation in the efflux pump do to the organism's antibiotic resistance? Reduces the resistance. It actually makes them more sensitive. In other words, one of the ways we could say this is that efflux pump has lost function. Okay? This is a scenario in which change in the structure, or change in the amino acid sequence of that efflux pump would lead to a change in the actual function of the efflux pump. Makes it work less well. It still works probably, but not as well. This idea right here is what some call evolutionary trade-off. So the idea behind evolutionary trade-off is that an organism can show an advantage in one of two conditions, but not both of them at the same time. In other words, Pseudomonas aeruginosa can show resistance to antibiotic, or can show resistance to phage OMK01. But it can't be resistant to both at the same exact time. Okay. And the reason why it can't be is because both of those pressures depend on the efflux pump. The phage relies on the efflux pump for entry, or for attachment, and the bacteria relies on the efflux pump to make it resistant to antibiotics. So, a long story short, they ended up using this treatment, and it worked. And they just within the past month or so actually published the case study of this particular report that you guys read about. But this is one area I think that phage therapy is going. Because we know bacteria will gain resistance to bacteriophage, and we saw an image where it can occur within 24 hours that we can select for mutants. The idea is, what if we can tailor what mutations are going to happen? What if we can select for mutations that actually make the organism, the bacteria, more sensitive to either an environmental condition or to an antibiotic, so that we can use the antibiotic and the phage together in synergy as a way to kill the microorganism. That's the design behind this. Okay. Any questions on this topic? I know it's kind of a big one. Allison.
What did you say the evolutionary trade-off was again?
So the evolutionary trade-off here, the efflux pump is crux for two possible outcomes. Phage require that efflux pump to gain attachment to the host cell. The efflux pump's also used for antibiotic resistance. So the bacterial Pseudomonas aeruginosa, choose is not the right word, but there's a selection for either become resistant to the phage or become resistant to the antibiotic. It can't be both at the same time, though. That's the trade-off, is that it can be one or the other. Okay? So the last thing to keep in mind here, just kind of as a way to bring this back to the holistic idea real quick, the main thing to keep in mind here is these effects are driven by mutations. These effects are driven by mistakes in DNA polymerase, okay, during the process of replication. So if you're looking for one connective thread between what we talked about on Friday, in resistance to gyrase, and what we've talked about today, resistance to bacteriophage, that's the common thread. Missense mutations are allowing for that resistance to occur. Okay? This is just one way in which we can use that resistance to our benefit. All right. If you did not pick up a handout from Friday, I'm just going to leave them here at the front. Feel free to grab one. Otherwise, I will see you all in lab tomorrow morning.